and welcome to this liver seminar Young Investigator edition. First of all, and on behalf of Professors Carmen Berasain and Matias Avila, and myself, Maria Arechederra, I would like to thank the organizers for the opportunity to hosting this April seminar. So it is my great pleasure to introduce our speaker today, Dr. Theo Heers. Theo earned his PhD in cell biology in 2017, studying prior diseases in the laboratory of Sophie Mouillet-Brichard in Paris. After that, he joined the functional genomics of solid tumor teams, led by Professor Jessica Zuckman-Rossi in the Cordelier Research Center in Paris. The main aim of the team is to study liver cancer genomics and the relevance of tumor heterogeneity and tumor microenvironment. In particular, Theo has mainly focused on the study of pediatric liver tumors. I would like to highlight today two of his more recent works. Theo was involved in the identification of a group of adult hepatocellular carcinoma driven by BAP1 alterations and harboring fibrolamellar-like features, and he also participated in deciphering the immune response induced by cisplatin-based chemotherapy in hepatoblastomas specifically developed in patients with German-line APC mutations. Nowadays, Theo is working on an integrated multi-omic analysis of a large cohort of pediatric liver cancers with a focus on identifying the mechanisms involved in tumor plasticity and resistance to chemotherapy. So before starting, I would like to remind the audience that at the end of the talk, Theo will take your questions, and we will also open the discussion time to our mentors, Professor Jessica Zuckman-Rossi, Carmen Berasain, and Matias Avila. So you can either write your question on the question and answer icon, or you can also raise your hand if you want to ask yourself. So Theo, thank you very much for accepting our invitation. And whenever you are ready, it's your turn.
Thank you, Maria. Hello, everybody. I'm really glad to give this talk, and I want to thank the organizers for this invitation. So today I will be speaking about the tumor heterogeneity and microenvironment in adult, but mostly in pediatric liver cancers. And so I work in the team of Jessica Zuckman-Rossi. I'm a postdoc, and so we work in the Cordelier Research Center in Paris, in France. So it's a really nice environment to work. And so we are in the team of Jessica Zuckman-Rossi, which is called FUNGEST, for Functional Genomics of Solid Tumors. And in this team, we work on 3 different types of tumors, liver tumors, but also mesothelioma and renal cancer. And we have different approaches to study the genomics of these tumors. And me, I mostly work with Eric Letouzé and Jessica for the computational analysis of genomics. So basically what we do is we use genomic data and bioinformatic tools to try to answer questions about clinical or biological or pathological features of liver tumors. And specifically, I try to study the tumor heterogeneity and microenvironment. So the tumor heterogeneity can take place at different levels. So first level is at the interpatients levels. So if you have patients with, for instance, liver cancer, you can try to separate them and find the different features of these tumors. And the goal of this is to do a good stratification of patients to find the prognosis, to give a different treatment, and maybe to use targeted therapy to treat specific groups of patients. So you can have different levels of heterogeneity. One of them that we study is the driver mutations of the tumors. So we have done in the lab studies of the genomic landscape of all HCC, so hepatocellular carcinoma. And we have also worked with focus on subgroups of tumors, for instance, tumors with a cyclin alteration or BAP1 mutated tumors. We also work with molecular classifications, for instance, with transcriptomic classifications. And if you study HCC, you probably know the different classifications, G1, G6, or Chung classification, or Hoshida. And we can also do a classification of tumors according to the DNA methylation. And there was a paper of a PhD student in the lab that was just out, and that I recommend you to read. And also, of course, specifically when you speak about liver tumors, we are interested in the background of the patients, because we know that there is a high heterogeneity of the etiology, the exposition. Most of the tumors are developed on a cirrhotic liver. And we can also study the germline predisposition of the tumors. And for instance, in the lab, we have worked on the integration of virus, AAV2 and HBV in HCC tumors. And also we have used the mutational signatures to study the process that give rise to mutations. And of course, all these different levels of heterogeneity have interactions between them. So there is a lot of things to study in the interpatient heterogeneity. But there is also another level of heterogeneity for a single patient. If you have a tumor, you can study, maybe if you have multiple tumors, you can study the different tumors. And also within a tumor, you can have samples at different regions. So this is a spatial heterogeneity. But also you can study the longitudinal evolution of tumors. For instance, if you have a first biopsy, then a sample from a surgery, and maybe a recurrent tumor. And then there is the heterogeneity at the level of single cells, because even in one sample, you will have different cells. And on top of that, you may want to study the microenvironment with infiltration of immune cells. So in the lab, we have done this for adult liver tumors. But today, I will mostly speak about pediatric liver cancer, because as Maria said, we have just a paper that was just accepted to study the integrated genomic analysis to identify driver genes and cisplatin-resistant progenitor phenotype in pediatric liver cancer. And so this is a big collaborative work with many contributors, and it was initiated in the lab by Guillaume Morcret, who did his PhD in the lab and brought with him as a subject of pediatric liver tumors. So I will just give a quick overview of the tumors, of the pediatric liver tumors. So the most frequent one is a hepatoblastoma. It occurs in really young patients, mostly before 5 years old. And it is the most frequent, but it is still a rare tumor. For instance, in France, we have around 20 cases per year. So this tumor is mostly sporadic, but in some cases, it is associated with some predisposition. And these tumors are highly heterogeneous, and they are developed on non-fibrotic liver. The current treatment is to use a first line of chemotherapy, then a surgical removal of the tumor, and then adjuvant chemotherapy. And the chemotherapy is based on cisplatin, and this treatment has a good survival of 80% at 5 years. Then you have pediatric hepatocellular carcinoma, pediatric HCC, that occurs later in life. In histology, they resemble the adult HCC, and they are mostly developed on pathological liver. And it can be with different etiologies, for instance, tyrosinemia or mitochondrial cytopathies, or in some countries it is linked with viral infection. And the treatment is a resection and an adjuvant chemotherapy, but there is a high level of chemo resistance and thus a poor survival of these patients. So third kind of pediatric tumor is fibrolamellar carcinoma, or FLC, that are developed in adolescents and also in young adults. They are developed on non-fibrotic liver, and they have a specific histology of the tumor cells, but also of the stroma with a lot of fibrosis in lamellar fashion. And finally, we have the pediatric hepatocellular adenomas. So those are benign tumors, and they are really rare and sometimes related to vascular abnormalities or genetic predisposition. So currently, there are a few genomic studies of these tumors, but not a lot. So for instance, in hepatoblastoma, there have been some studies of whole exome sequencing, and the results have shown that there are few mutations per tumor, and also only a few drivers have been described, and mostly mutations in CTNB1, which is beta-catenin. For the HCC, there is only one study with whole exome sequencing and one study with target sequencing, and they have found some alterations in the Wnt pathway, but they did not identify drivers in HCC with underlying liver disease. Then for the FLC, it is really well characterized because there was a paper in 2014 showing that there is a recurrent driver fusion that is found almost in all FLC tumors. And it is a fusion between DNAGB1 and PERK-ICA that gives rise to the activation of PKA. And then for the adenoma, to my knowledge, there is no genomic studies of these tumors. So the first question that we wanted to ask is that we know that there is some overlap in the age and also of the histology of those tumors. So we wanted to see if there is a continuum, a molecular continuum between the tumors, or if we can see a link to the age. And then we also asked questions more focused on hepatoblastoma, because we know that hepatoblastoma has a really simple genetic landscape, only few driver mutations, but they have a high phenotypic heterogeneity at the histological level. You see here examples of the various histology that the pathologist can see in hepatoblastoma. And also at the molecular level, there have been a lot of transcriptomic or methylation classification despite the simple landscape. So we wanted to know what drives the heterogeneity of hepatoblastoma, and this will be the second part of my presentation. And also we know that a few patients will resist to chemotherapy, and there is only limited therapeutic alternatives for now. So we want to know what are the driving forces of resistance to chemotherapy, and see if it is possible to use targeted therapy to overcome this resistance. So to answer these questions, we studied a multi-omics. We use multi-omics to study a cohort of pediatric liver tumors. We work on frozen tumors with HCC, FLC, HCA, but mostly on hepatoblastoma, where we have 65 patients and 104 samples. And specifically, our cohort has patients with poor outcome. We have samples before chemotherapy, after chemotherapy, and also we have samples of relapse and metastasis. So this allows us to study the heterogeneity. And on this cohort, we applied the different omics, so whole genome sequencing or whole exome sequencing, and also RNA sequencing and RBS, which is a method for the methylome. So now I will go to the results. So I will start with the driver landscape of pediatric liver tumors. So in the tumors that we studied with whole genome sequencing, so we had 65 samples. You can see here for all these samples, the number of mutations that we found and also the number of structural variants. And it is classified with the different diagnoses. So the first thing that we observe is that, as expected, the hepatoblastoma have a low mutation burden, a low number of mutations. But then in the recurrent hepatoblastoma, so the relapse or metastasis, we have a really higher number of mutations in these tumors. You can also see that hepatoblastoma have only a few structural variants, except for a small subset of older tumors with complex events, such as chromoplexy. But then here you see that the pediatric HCC has a really different profile of structural variants, because they all have a lot of deletions. So there is a deleter phenotype with a median of almost 50 deletions per tumor. These are small deletions. You can see here on this example, this is a chromosome 2 of HCC tumor. And you see here the different deletions that are really small. And what is interesting is that the HCC that we study have various etiologies, and still they all, almost all, show this deleter phenotype. So we don't know currently what is the cause of this phenotype, but this is a recurrent phenotype in these tumors. So then, if we expand the cohort to the patients with whole genome or whole exome sequencing, so we have 84 patients and 122 samples. And so here, this is a landscape of mutations for the hepatoblastoma, adenoma, fibroadenomatous carcinoma, and HCC. And you can see that the Wnt pathway is altered in most hepatoblastoma cases, 98%, but also in some adenomas and in HCC. So for hepatoblastoma, almost all patients have a mutation or a deletion of beta-catenin. And you can see that the few patients that don't have a mutation of beta-catenin have a germline alteration of APC or AXIN1. And in the end, there is only one patient who doesn't have a mutation in the Wnt pathway. For the pediatric HCC, we don't have any mutation of beta-catenin or APC, but we have alteration of AXIN1, which is part of the destruction complex, and also of AMER1, which is another part of the destruction complex of beta-catenin. And what is interesting is that these alterations are not through mutations, but through deletion. So homozygous deletion or deletion with truncative mutation. And this is related to the deleter phenotypes that I just showed you. The second pathway that is highly altered in these tumors is 11p15 locus. So this locus is an imprinted complex locus on chromosome 11. So normally each cell has two chromosomes. The paternal chromosome is linked to proliferation and the expression of IGF-2, while the maternal chromosome has an anti-proliferation role and is linked to the expression of CDKN1C. And there are two spots of methylations that are differentially methylated between paternal and maternal and control the expression of the genes of the locus. So in hepatoblastoma, you can see a lot of alterations of this locus through different ways. So mostly it is through a loss of heterozygosity, or LOH, where you will lose the maternal copy, and sometimes you will gain a second paternal copy. So it is a copy neutral LOH. But we also found through analysis of methylation, patients or tumors with an alteration only of the methylation of this region, or a loss of methylation in this region. And finally, for a few hepatoblastoma patients, we found a truncating mutation of CDKN1C. And so this pathway is altered in most hepatoblastoma, but also in hepatocellular carcinomas. And actually, when studying this alteration in hepatoblastoma, so this is the work of Gilles Pillet, who is a PhD student in the lab, we observed premalignant clonal expansion of cells in the non-tumor liver that do have the alteration of the 11p15 locus. For instance, this is a hepatoblastoma patient. So this is the BLL frequency profile in the tumor for the chromosome 11. You can see here that you have a loss of the heterozygosity within the tumor. And then when we look in the non-tumor liver sample, we also find this alteration. So it means that this alteration is already present. So this is a copy neutral LOH of this locus. It is already present in the non-tumor liver. And actually, we found this pattern in 6 patients. So it is 10% of hepatoblastoma, where we have different level of mosaicism in the non-tumor liver. And then the tumor will expand from one of these cells that will also get a mutation in beta-catenin. So another way to visualize this is to look at the expression of IGF-2, which is controlled by this locus, in the non-tumor liver of samples without the mosaic, and these 2 samples with a mosaic alteration, where they express high level of IGF-2 that are similar to tumors. But then if we look at markers of the beta-catenin activity, for instance, AXIN2, you can see that it is increased in the tumors, but it is not increased in the non-tumor liver. So the non-tumor liver has the alteration of 11p15, and that can be a predisposition to a further transformation by the acquisition of a mutation in beta-catenin. And another way to visualize this is still the work of Gilles Pillet. She did some RNA-scope experiments to see the expression of the IGF-2 gene. And so here in this same patient, you can see that IGF-2 is expressed both in the tumor and in the non-tumor liver, while when we look at markers of beta-catenin, they are activated in the tumor, but not in the non-tumor liver, where you see the normal zonation. And in another sample, another patient with no mosaic alteration, IGF-2 is not expressed in the non-tumor liver. So this is maybe this expands the number of tumors with a predisposition, because we found that 10% of hepatoblastoma have these mosaic alterations. So going back to the landscape of drivers, I just showed you 2 drivers with high frequency, but then we directly go to drivers with low frequency in hepatoblastoma or HCC. And so we found a high number of these drivers, but with always low low frequency. And what is interesting is that we have different drivers where we find an alteration through a mutation in hepatoblastoma, And then in HCC, we found alterations of the same gene, but through a homozygous deletion. And this is linked to the deleter phenotype of these tumors. So it shows that both diagnoses will alter the same pathway to be transformed, but through different mechanisms. Also, among the different genes that we found altered in these tumors, we found some genes that could be targeted in patients, for instance, RPS6K3, which is altered in some hepatoblastoma through mutations and some HCC through deletions. And what you can also see is that while hepatoblastoma and HCC share a lot of mutations, the adenomas and fibrolaminar carcinoma have a really distinct driver landscapes. And finally, for the drivers, we have found a relation between the type of alterations and the age of patients. Because, for instance, for beta-catenin, in young patients, we found mutations, mostly mutations, while in older patients, beta-catenin is always altered through deletions of the exon 3 or exon 3 and 4. And we have same relations for TERT, 11p15, and in general, the number of drivers, which is higher in older patients. So now, moving on to the transcriptomic classification of hepatoblastoma, we have performed a clustering on RNA-seq data from 100 hepatoblastoma samples from 64 patients. And we have found three main groups of tumors. So this group that we call hepatocytic hepatoblastoma is related to the expression of markers of hepatic differentiation, and it is enriched in fetal histology. We have a second branch here that we call liver progenitor that express a lot of progenitor markers, and it is linked to the embryonal histology of tumors. And then we have a third group, the mesenchymal group, which expresses mesenchymal markers and is related to a mesenchymal histology. So adding to this differentiation, we also find a link to the level of immune infiltration, because if you look here at the expression of genes related to immune cells, you can see that it is different between the branches. For instance, the liver progenitor branch has a low level of immune infiltration. You can also see this here. The mesenchymal branch has an intermediate level of immune infiltration. And then in the hepatocytic tumors, we have two subgroups of tumors that we have called hepatocytic hot and hepatocytic cold. And if we just restrict the analysis to these hepatocytic tumors, we can see that the samples with chemotherapy have a higher level of immune infiltration compared to samples with no chemotherapy. So probably there is a priming role of the chemotherapy to induce the immune response, but only in the hepatocytic parts of tumors. So we can study the heterogeneity of tumors because we have 24 patients with multiple samples sequenced. And so here, for each line, you have one patient with its different samples. And you can see here some patients, all the samples are in the same branch, but also we have some patients with phenotypic switch. For instance, this patient, this is an example of spatial heterogeneity because we have two samples of the same primary tumor. So one sample here, it was classified as hepatocytic cold, while the other sample was classified as liver progenitor. And what is interesting is that we have a difference in phenotype, but the two samples have the same driver alterations in 11p15 and beta-catenin. So this is an example of plasticity because with the same drivers, we have two phenotypes. Here, this is an example of longitudinal heterogeneity, because here we have a biopsy of a tumor before the chemotherapy, and it is classified as hepatocytic cold. And then the other sample after the chemotherapy is classified as mesenchymal, and actually you can see here the osteoid differentiation. And in this case, the two samples have the same beta-catenin alteration, and the 11p15 alteration is only found in the biopsy. And then a last example of longitudinal heterogeneity. This is a patient where we have studied the primary tumors and different samples of the relapse or metastasis. And so in this case, the primary tumor is classified as hepatocytic hot. And then this is a sample of metastasis that developed on the grafted liver, and it is classified as a liver progenitor. And once again, both samples have the same driver alterations. So we have a lot of heterogeneity, spatial and longitudinal, without necessarily having a change in driver. So we show that there is a high plasticity of hepatoblastoma cells. We have also studied the methylation of the tumor. So this is the work of Amélie Rohring, who is a PhD student in the lab. So we have used reduced representation bisulfite sequencing of 84 hepatoblastomas. And Amélie has performed a clustering of the tumors, and we found 4 groups that are related to the differentiation. So we have hepatocytic, liver progenitor, and mesenchymal, but also a group which is related to the age of the patients. You can see here that these patients are older. And so we found a relation between these groups of methylation and also the group here of transcription. So Amélie has also performed an analysis, an independent component analysis, to find some scores that correspond to the contribution of different processes to methylation. And here, this is a representation of 2 of these components. And so this component is related to the histology, fetal or embryonal, and this component is linked to the mesenchymal pattern. And you can see the colors here are linked to the transcriptomic groups, and that we have a good concordance between the transcriptomic groups and the methylation profile. And for patients where we switch groups, you can see that we also switch group in methylation. So it tells us that the change that we see in transcription and so those different groups are related to the epigenetic status of the hepatoblastoma cells. And if we go into the details of the methylation, globally, the mesenchymal hepatoblastoma have hypermethylation of HNF1A, HNF4N, and PPAR gamma, while we have also changes that are related to the liver progenitor hepatoblastoma or the hepatocytic hepatoblastoma, and also another level which is related to age. Another way to study the heterogeneity is to go to the single cell level. And so for this, we used a single nucleus RNA-seq to study 3 frozen samples from the same patient. So we have 1 sample from a non-tumor liver, 1 sample of the primary tumor, and also a sample of a metastasis developed in the lung. And so this is also an analysis of Amélie. And she used a virtual copy number alteration to assess the copy number through the expression data. And so thanks to this method, she can restrict the analysis only to tumor cells, where you can see here the gains on different chromosomes that is inferred by this method. And so when looking at only tumor cells, we find that we can identify three different types of cells that are corresponding to the three groups that I presented in bulk analysis. So for instance, in the primary tumor, we have here cells corresponding to mesenchymal differentiation, here cells that are more of the liver progenitor groups, and then here these cells are, correspond to the hepatocytic differentiation. And we found this diversity of cells both in the primary tumor and also in the metastasis. And in this patient, we have found that the metastasis has a specific alteration, a gain of chromosome XQ. So we know that all samples from the metastasis derived from a single sample that got this alteration, and still we found the different groups in the metastasis. So the hepatoblastoma cells were able to create the diversity of the different groups, both in the primary tumor and then in the metastasis. And it is another way to illustrate the plasticity of hepatoblastoma that is probably driven by epigenetic mechanisms, since it is not related to driver alterations. So now we go to analysis of cisplatin resistance, and for this we use the study of mutational signatures. So it was performed by Benedict Montero, who did his master in the lab. So mutational signatures are it's a method where you study all the mutations in the tumor, and you look at the context, the trinucleotide context of each mutation to categorize them. And with this, you can find some mutational signatures that are like the genetic footprints of the process that give rise to mutations. And so in hepatoblastoma, we identified 4 signatures of single base substitution. You can see them here, SBS1, 5, 18, and 35. And I will mostly focus on SBS 35 because it was already shown that it is linked to cisplatin adducts. And actually, in the lab, Jill performed some experiments where they exposed cell lines to cisplatin, pediatric liver cancer cell lines to cisplatin, and then she was able to retrieve these signatures in the cell lines with the cisplatin treatment. You can see here the 2 specific red peaks that are the signatures of cisplatin. So in hepatoblastoma, we found this signature, but where exactly do we find it? So this signature is never retrieved in samples before chemotherapy. This is expected. But then in post-chemotherapy primary tumors, we found this mutation, but only in a subset of tumors. And then in recurrent tumors, relapse or metastasis, we find a huge number of clonal SBS35 mutations. So it was subclonal in the primary tumors, but it became clonal in relapse and metastasis. And this means that all the recurrent tumors are clonally derived from a cell which accumulated thousands of SBS35 mutations. If you take, for instance, this patient, in her biopsy before chemotherapy, we don't find these mutations. But then in the primary tumor, we find more than 1,000 of these mutations, but subclonal. And then in 2 metastases, we found thousands of subclonal and clonal mutations linked to cisplatin. And if we go back to the primary tumors, I told you it is only found in a subset of tumors. And actually, if we look at the transcriptomic classification, you can see that these mutations are always retrieved in tumors from the liver progenitor groups, but not in the other groups. So it means that the liver progenitor cells are the ones that accumulate cisplatin mutations, so they can resist to cisplatin, and they are at the origin of the relapse and metastasis that will later have plenty of these mutations. And for instance, if you take this patient that I showed you earlier, we have two samples from the same tumor. So both samples had the same chemotherapy regimen, but in the hepatocytic cold sample, we don't find any of these mutations, and we find more than 6,000 of these mutations in the liver progenitor sample. Another way to visualize this result is to look at the phylogenetic trees of patients of tumors where we have both primary and recurrent samples. So here, each tree is one patient, and the size of the branches is related to the number of mutations. And in each case, we have the same scheme. So for instance, in this patient, in the primary tumors, we have only a few mutations. And then in the metastasis, we have accumulated thousands of mutations. And the color of the branch is related to the type of mutational signatures. And so this blue color that you find in every relapse or metastasis corresponds to the accumulation of thousands of mutations that are linked to cisplatin. And so this signatures tells us that a cell was able to proliferate and accumulate this mutation during the treatment. And actually, we find that the presence of this signature in the primary tumor is associated to a bad prognosis in terms of progression and of survival. So we would want to target the cells that are resistant to cisplatin. And since we show that the liver progenitor cells are the ones that are resistant to chemotherapy, we looked for genes that are commonly overexpressed in liver progenitor hepatoblastoma compared to other tumors. And we identified 3 candidates that could be targeted. So there is BRCA5 or Survivin, PLK1, and CHEK1. So to study these genes as a candidate for therapy, we used pediatric liver cancer cell lines. So we have 9 cell lines, 8 of hepatoblastoma and 1 of pediatric HCC. And actually, these cell lines have the same driver alterations that we found in the tumors. So this is, again, the work of Gilles Pillet in the lab. So what you can see here is that if we treat these cell lines with cisplatin or carboplatin, most of them are resistant to cisplatin. And you can see here the purple color is related to a score of liver progenitor. And the more resistant cells are the ones that have a high liver progenitor signature. And then if we treat these cells with an inhibitor of our 3 candidates, you can see that the cells have better sensibility than to cisplatin, and also that cell lines with a high purple color, so a high liver progenitor signature, still are able to respond to these drugs. So finally, we did a xenograft experiment with the HepG2 cell line. So it was a proof of concept to validate the best candidate, which is the inhibitor of PLK1. And so in these experiments, mice were injected with HepG2 xenografts, and the xenografts were able to grow even with cisplatin or cisplatin and doxorubicin, but they were sensitive to this inhibitor of PLK1 despite their resistance to cisplatin. So this is a proof of concept in vivo that PLK1 could be targeted to overcome the resistance to cisplatin. So now we'll just go through my conclusions. I see I'm a little late. So what I told you today about pediatric liver tumors is that for their drivers, we have non-alterations of Wnt beta-catenin, but also the 11p15 locus is altered in more than 80% of hepatoblastoma and HCC. And we have found common pathways that are altered in both hepatoblastoma and HCC, but through different mechanisms. And we have identified new drivers, including targetable genes. I have showed you that we have a molecular classification of hepatoblastoma that is related to the differentiation, but also to the immune infiltrations. And there are a lot of phenotypic switches, spatial and longitudinal, between the groups, but with no change in drivers. So there is a high plasticity of the hepatoblastoma cells, and we validated this plasticity in single nucleus experiments. And finally, for the resistance to cisplatin, we have shown that during the chemotherapy, the liver progenitor cells will accumulate subclonal cisplatin-related mutations, and then the relapse and metastasis are clonally derived from these cells that have accumulated thousands of these mutations. And we have studied 3 candidate drugs that could be used to specifically target this liver progenitor phenotype and bypass the resistance to cisplatin. So thank you everyone for your attention, and I also want to thank my team and all my collaborators, and I thank once again the organizers for the invitation.
Okay, so thank you, Theo, for this nice presentation and for sharing with us all this huge amount of work on hepatoblastoma. And before starting with the questions, as I said at the beginning, for the discussion time, we have also our mentors. So I would like to welcome Professor Jessica Zuckman-Rossi. Thank you, Jessica, for being here with us today.
Hi, thank you. Thank you for the invitation. It's a pleasure to be here.
And I also want to welcome Professors Matías Ávila and Carmen Berasain.
Yes, so it is a true question, because, for instance, we have seen for some tumors, the pathologist has only seen a small amount of the histology which is associated to the bad prognosis, but still the sample was taken from this small part of the tumors, and the phenotype was the aggressive type of liver progenitor. So it means that if you take a sample in a small part of the tumors and you use it to classify the tumors, you will have maybe a false impression of the tumor. So this is a really good question, but there is no easy way to answer. One way would be to always take multiple samples of a tumor to study them, to be sure to not miss anything, and maybe then look at the worst case scenario. And maybe another way is to go back to the histology of the tumors, because the histology is able to give an impression of the whole tumors. And if we can have a good correlation between a molecular finding and the histology, then we can use the histology to classify the percentage of cells, for instance, of the liver progenitor phenotype.
So maybe if I can continue on this line, I think that heterogeneity in hepatoblastoma and liver cancer is more important than in adult liver cancer, in our experience. But it enlightens also the necessity to translate the molecular classification in imaging, not only for the pathologist, but you need to have the whole tumors to translate it, but also in radiology. And our hope for the future is also to assess the heterogeneity and the identification of the different subclasses of tumor with imaging. It has been already done for the classification of adenoma, for example, that are very simple because it's benign tumors. But we hope also that in the future, it will be also the case for malignant tumors, in pediatric, in children or also in adults. And but it's required to analyze a large series of patients with having both the molecular classification and the imaging, and then to be really prompt to translate the knowledge into the clinics.
Yes, so this is actually what we see, because if we take our classifications, we see that the liver progenitor cells, so the ones that are more aggressive, are also cold tumors. So, and in hepatocytic tumors, we see a lot of immune infiltration, mostly after the chemotherapy, while in the liver progenitor cells, even after chemotherapy, there is no immune infiltration. So there is really a link between the cell type and the immune infiltration, but we are still we still need to study to understand what drives the immune infiltration, and if there is an active immunosuppression by the liver progenitor cells, or is it the hepatocytic cells that will attract the immune cells. But yes, there is a high link. So we have not done this. Maybe we should ask Jessica if she wants to perform this. But
that's a very good question. It's a question of money, not only of money, but also of techniques. Recently, there is a huge progress in the technology for proteomic analysis. And we will try to go directly in the spatial distribution and to look at the heterogeneity on the slides with some new techniques that are developed by NanoString with the DSP system, then can assess both the transcriptomic profile and the proteomic profile in the same slide, and to see what are the interplay between the different contingent of the tumors. It's very expensive, I have to say. But I think that also the model of hepatoblastoma is a very good model to study this interaction, because it's more pure than in adult tumors. And we have this contingent and this area of tumors that are purely progenitor or pure, or not exactly purely, but highly enriched in liver progenitor or highly enriched in hepatocytic molecular profile.
So yes, I think in general, the liquid biopsy is a good way to study the longitudinal the longitudinal evolution of a tumor, because once you have first diagnosed the tumor, you can know the mutations and then just follow this mutation along the time. And also through liquid biopsy, maybe you can also assess the heterogeneity, because you will have access to mutations that maybe were not present in the specific sample that you took from the tumor, but that will be present in the blood. So yeah, I think so.
Okay.
But we have no experience at this moment.
Yes. So I cannot say that there is a specific epigenetic alteration that will be responsible for the resistance, but what we see is that the liver progenitor cells are the ones that resist. And when we studied in more details the mechanisms of resistance, we found the overexpression in these cells of a pathway of DNA repair. So probably these cells are able to overcome the cisplatin adducts on DNA by repairing the DNA, and maybe this is why they accumulate so much mutations. And what we have found is that some of these genes involved in DNA repair are also highly expressed in fetal liver cells. So maybe this liver progenitor phenotype is actually mimicking a phenotype of liver development, so of progenitor cells that have also the intrinsic ability to resist to DNA damage. So maybe it's an epigenetic change, but not a specific epigenetic alteration, but more the epigenetic landscape that will put the cell in a progenitor state that also is related to the resistance to cisplatin. So yes, so this is because we work with frozen tissue. This is why we went with single nuclei and not single cell, because when you have frozen tissue, it's hard to separate the cells. But we don't have a lot of experience for a single cell in the lab. So we did some experiments with single cell in cell lines. And I think this was our first experience with tumors. So it worked. And actually, I don't know the details for the number of reads per nuclei. Yes, it is. It is related. So in the same tumor, sometimes we have two or three samples in the same tumor and the same surgery, and the immune infiltration is really different, and it is related to the cell state because the liver progenitor cells are really immune cold. Yes, so we have seen two aspects of chemotherapy. For the classification, we see more mesenchymal samples after chemotherapy. Actually, we never see a mesenchymal classification before chemotherapy. So maybe there is a role in chemotherapy to pushing cells towards the mesenchymal differentiation. Also, we have not quantified this, but we can imagine that chemotherapy will select for the liver progenitor cells because we show that they are the ones that are resistant. And then at the level of immune infiltration, we see that in the hepatocytic type of cells, the immune infiltration is higher after chemotherapy than before chemotherapy. So maybe there is an immune related death, an immunogenic cell death that could be priming the cells to be recognized by lymphocytes and triggers the immune response.
And just to complete, it was excellent, Theo, but something that is interesting also for the people that are working with adult tumors. Remember that the liver progenitor compartment of the tumor is those that accumulate tons of mutations, and they are cold without immune response after chemotherapy. So just to open also that we were supposed to have all the neo-8p-tops and most of the mutations that can be also deleterious translated in abnormal peptides are those that are cold in our in hepatoblastoma after chemotherapy, and that was very surprising.
Okay, thank you. So before ending, I would like to thank again Theo and Jessica for being here with us today and for sharing this huge amount of work. It was really, really nice. And thank you, Carmen and Matthias, and also the organizers, Rafa and Jordi. And thanks all the audience for your attention. So bye-bye.
Bye-bye. Thank you very much.
Bye. Thank you. Bye.